Good morning. My name is Richard Vaughan-Jones. I'm the president of the Physiological Society, and it's my great pleasure to introduce this morning's lecture, which is a prize lecture, one of the very distinguished lectures awarded by the Physiological Society, and it's the Hodgkin Huxley Katz prize lecture. This is awarded every other year to a distinguished physiologist, usually in the field of biophysics, membrane biophysics in particular, for a person who is not from the UK or Ireland, so from Europe or overseas. And I'm really delighted to introduce today this year's winner of the prize lecture, which is Professor Karin Sipido. And Karin is an international expert on calcium handling in the heart, particularly in clinical conditions such as cardiac hypertrophy and heart failure. She's a professor and head of division of experimental cardiology in the Department of Cardiovascular Diseases at the Catholic University in Leuven in Belgium. And Karin's an important example to all of us of a clinician who crossed over to the dark side, and by that I mean basic sciences, and then learnt a lot about mechanistic events that are occurring in the heart, but is also now crossing back, and by that I mean entering into the field of translational medicine. So she's linking classically basic sciences with translational clinical work. So she's interested in calcium signaling, the mechanisms, and then understanding how those mechanisms can become drivers of cardiac disease. She did her medical training at the University of Antwerp in Belgium, specializing in cardiology, and there she actually combined her training with some research experience, and that fired her up. And then she went to the United States to do some research, and her research training really was done backwards, in that she appears to have done her postdocs first and then her PhD training afterwards. By that I mean that she was a research fellow in the University of Maryland in Baltimore in the laboratory of Gil Weir, and then subsequently in the laboratory of Eduardo Marban at Johns Hopkins, also in Baltimore. So I would classify that as her training, but it's sort of postdoctoral training. There she learned about calcium signaling and basic mechanisms, but then transferred back to Belgium, to the University of Leuven, to Edward Karmelit's laboratory. Now, Karmelit was the father of quantitative electrophysiology in Europe. So that then gave her a good fundamental training in electrophysiology, and it's there that she studied for a PhD. She then stayed on as an academic member of the faculty and became a full professor in 2002. She's the co-founder and chair of the Council of Basic Cardiovascular Sciences, and has had numerous plenary and prize lecture awards. I won't be enumerating them here. But she's been on the editorial board of the Journal of Physiology and of Circulation Research, and she's currently the editor-in-chief of Cardiovascular Research. And I can say that she's known to all of us in the field for her elegant and rigorous approach to her science, in particular the role of membrane sodium-calcium exchange in the failing heart. So we look forward, Karin, to your lecture today, which is entitled Calcium Microdomains in Cardiac Myocytes. So please welcome Professor Sipido.
Thank you, Richard. Can you hear me? Is this microphone working? Yeah. I'm very honored by the invitation and pleased to be here. This is a named lecture named after the pioneers in electrophysiology in excitable cells. And you may have wondered why this topic and why this person. Richard partially explained it, but let me take you a little more into detail. When I started as a student with Dirk Brutzart, we worked on cardiac physiology. Electrophysiology was rather far away. It was the current that was needed to stimulate the papillary muscle in the mouth. I then went to Gil Weir, where I started voltage clamp, and so I really measured ionic currents, but mostly limited to the L-type calcium current. It was Eduardo Marban who introduced me to the more fine detail and really into the topic of ion channel biophysics. And within the first 24 hours that I was in his lab, I had on my desk Bertil Hille to read and the 3 landmark papers by Hodgkin, Huxley, and Katz, with a questionnaire that I had to fill out, and I was quizzed afterwards whether or not to test that I had read everything in detail and knew all there was to know. Then I continued, as Richard said, my work with Edward Carmelit, where indeed I continued in electrophysiology and learned about the different membrane currents in the heart. That took me to my work in the lab on calcium and arrhythmias, combining also with the clinical training I had. There is another part to the story, which is in 2001, when one of my students, Frank Heinzel, started to work on the calcium microdomain, and I will show some of his work here, and we came together to the FISOC meeting in Oxford. And at the breakfast table in the morning, we were sitting next to this gentleman, and Frank, being very polite, introduced himself first and said, I am a student with Professor Sipido in Leuven. I work on calcium, and may I inquire about your research interests? So the gentleman, being a gentleman, said, I'm Andrew Huxley. I worked in electrophysiology, ion channels, axon squid, and so on. And we had a very pleasant conversation that ended with a dinner in the evening. And I think it really marked both Frank and myself for our further work. And this was my last and close encounter with one of these pioneers. So in my talk, I will introduce first the concept of a calcium microdomain. How did it come about? How much calcium is there in this microdomain? And is there a relation, what kind of relation, to not only calcium, but sodium through sodium-calcium exchange? And in the last part, I will talk about microdomains around ryanodine receptors and remodeling in disease. This is where it all started. Excitation-contraction coupling in ventricular myocytes was defined in the late 80s using the techniques that were then developed of the voltage clamp, the fluorometry, the measuring of the global cytosolic calcium, and the single myocytes. And here you see the amplitude, the amplitude of the calcium transient as a function of the polarizing voltage steps, and below it, the amplitude of the L-type calcium current. And you can see there's a close match with a bell-shaped relationship. So calcium release from the sarcoplasmic reticulum through the ryanodine receptor is graded by the trigger calcium influx. Now, as this is a calcium-activated channel, there is a paradox here. How can it be that there is such a tight coupling? Why is this not an all or none phenomenon? Because as soon as calcium is coming out of the sarcoplasmic reticulum, there should be a further activation of all the RYRs, all the ryanodine receptors, and this should be an all or none phenomenon. This was addressed in computational modeling in another landmark paper by Michael Stern, who examined several models of interactions between the L-type calcium channel, LTCC, and the ryanodine receptor, and taking into account that these are channels with stochastic behavior, that they would be interacting in close proximity with a restriction of calcium diffusion, he could explain this graded behavior. And it required then the concept of a microdomain of interactions. So this is our current view of excitation-contraction coupling. It occurs in a restricted space that was also, thanks to the work in electron microscopy at the same time, defined in size. So this is the T-tubule, schematically shown here, with next to it the sarcoplasmic reticulum, and in between the dyadic cleft, where you have an interaction of L-type calcium channels and ryanodine receptors in close proximity. During the action potential, we have activation, depolarization, opening of L-type calcium channels. They activate a ryanodine receptor. This is the major source of calcium, which rises highly then in this microdomain. And then the calcium activates the contraction. Included in this area is a sodium-calcium exchanger that is responsible for calcium removal, that comes in through the L-type and maintains the balance. And then there's also the sodium handling that may also affect the activity of the sodium-calcium exchanger. So in that dyadic cleft, how much calcium is there, given that this is a very restricted area? The first answers came from computational modeling. This is an example of a diet here shown the T-tubule membrane. This is the junctional sarcoplasmic reticulum. This is the ryanodine receptors protruding into this narrow space, which is about 12 nanometers in height. And then lots of parameters were entered to look at the actual changes in calcium. The opening of the L-type calcium channel gives rise to an increase in calcium of about 1 micromolar within a radial distance of 15 nanometer, the RYR cluster area, in less than half a millisecond. So quite high values came out of that. But even higher values came out of the calculation of the contribution of the opening of the ryanodine receptor, which went up to several hundreds of micromolar at the center of the cleft, and even up to 100 micromolar at the periphery, around 20 milliseconds. There's been quite a bit of debate and improvement also in the models by many people, including Mark Cannell, Michael Sturm himself, Eduardo Rios, and others. But it's always remained in very high values that exceeded by 2 orders of magnitude the calcium that is typically measured with a cytosolic calcium reporter dye. So can we get an experimental validation of these computational values? The way people have addressed this, including ourselves, is to use the reading of calcium-dependent currents. The channels within the dyad, the L-type calcium channel, is actually quite sensitive to the calcium near to it, and it responds by closing or better inactivating. And the sodium-calcium exchanger as well is modulated by the prevailing calcium. So can we use these as reporters for the calcium in that cleft? Here is an experiment that we did a number of years ago, where we measured the L-type calcium current here and in green dotted on the right during a depolarizing step to minus 35 millivolts. And you can see this hump on the current, which is actually inactivation and recovery from inactivation. And that is confirmed by looking at the available current during a step to 0 millivolt. Below here is the calcium cytosolic reporter. Basically, you don't see anything because the cell is dialyzed with EGTA. Nevertheless, there is this clear inactivation which must result from the microdomain interaction of local calcium on the LTCC. We then went on with another experiment where we again looked at the degree of inactivation of the L-type calcium current by increasing amounts of calcium release from the sarcoplasmic reticulum. We applied a calibration. We did in situ in the cell. We examined the relation between the amount of inactivation and calcium in a steady state, and applying that to these transient values, this is what we came up with. What you are looking at here is a profile of the L-type calcium channel inactivation in time. So it peaks very early and then recovers. And on the y-axis is the calibrated calcium values that it is reporting on. So we find a value of 10 to 15 micromolar. It's independent of the number of activated release sites. It's really talking about dyadic cleft calcium. So this is much less than the theoretical predictions. Why? We can only hypothesize here. It's possible that we should not use the total L-type calcium current because there may be a number that is not in dyadic cleft. That's usually estimated at less than 10%, but nevertheless. It's also quite possible that the indicator we are using is not optimal and may be saturated and not telling us the real maximal values. We did a similar kind of exercise using the sodium-calcium exchanger. And the sodium-calcium exchanger, it is known that it is rather distributed and that some of the sodium-calcium exchangers are sensing a much higher value during release. And this was first proposed by Andy Truffert and David Eisler in 1995. And this is a repeat of their experiment. This is caffeine inducing calcium release from the sarcoplasmic reticulum. This is the accompanying sodium-calcium exchange current. And here the current is plotted as a function of the calcium. Calcium here measured in the cytosol by fluO3. This is the hysteresis that you see during the release of calcium. So during the upstroke here, you have a much larger amount of current than when you look at the current in relation to the cytosolic calcium, later on indicating that there is actually a much higher value near to the sodium-calcium exchanger at that time. We did another type of approach, and this is the work of Karoli Aksai when he was in the lab. And so what he did, and I'll spare you the details, this is a depolarizing step with the cytosolic calcium transient, and this is below in black the sodium-calcium exchange current recorded at this point in time. And he then extracted two components. One that was the current for the global calcium in green here, and based on the modeling of the cytosolic calcium, and the remainder current then can be ascribed to the local calcium. From that, we reconstructed a profile of dyadic calcium using this two-compartment fitting with maximal values around 10 to 15 micromolar. So using a totally independent measure, actually coming up with very similar values as what we had for the L-type calcium channel. Nevertheless, it's much less than what is taught in the rigorous computational model, and maybe we have to think even more compartments. And on the right here are the values for what is the global calcium, which is around 1 micromolar, the subsarcolemmal, which could be several micromolars but less than 10, and then in the real cleft, like very, very close to the ranitidine receptor, maybe it goes up to 100, but further modeling also indicates that then we are talking about very few molecules in a very, very small space. So what about the relation to sodium and sodium-calcium exchange? So the sodium-calcium exchanger responds to calcium by increasing inward current and calcium removal, but with an increase in sodium, it's going to go into reverse mode and bring in calcium. And the first paper on the inclusion of sodium-calcium exchange in this microdomain was in 1991 by Leblanc and Hume, with a comment by Nigli and Lederer, who launched the concept of the fuzzy space. And the observation and the interpretation was that activation of the sodium channel down here would drive sodium to very high levels in a small space that would then have the sodium-calcium exchange go into reverse mode, bring in calcium, and activate the ranitidine receptor directly without L-type calcium channel involved. Now, that led to a very nice scientific debate with a lot of thunder and lightning, with opponents and proponents, and a lot of papers. I'll show you only two of them, one that we did ourselves, where we looked at calcium entry in the diet via reverse mode sodium-calcium exchange and concluded that it is a poor trigger for ryanodine receptors because the flux through a sodium-calcium exchanger is much lower, so you have a much less, smaller and slower rise in calcium to activate the RYR. If you just focus on this part of the records, you see the calcium transient evoked in the presence of nisoldipine. With or without, you have a slow but a large calcium transient. But if you look at plus 10 millivolts, which is a typical plateau action potential value, then you see that sodium-calcium exchanger alone is a very poor trigger. And we did some further computation on that. Now, this is a sort of compromise or comprehensive view coming from data in the sodium-calcium exchanger knockout mouse and a lot of the work of Josh Goldhaber together with John Bridge and Ken Phillipson. And here the current view is that calcium entry into the diet via reverse mode sodium-calcium exchange is like contributing to the triggering of the L-type calcium channel, but quantitatively we don't know exactly at this moment how much it is contributing. This is not a closed debate. It's actually kind of reopening as people are now looking at the role of sodium entry in determining calcium in the diet and thereby the activity of ryanodine receptors. So for the last part of my talk, I'd like to look at the ryanodine receptors clusters themselves. They are sitting as a sort of microdomain that themselves, they have a lot of signaling molecules close to them. They are sensitive to phosphorylation by cyclic AMP, that is modulated by PDEs, phosphorylation through CAM kinase, modulation by oxidation, nitrosylation, luminal calcium, and there's a lot of signaling molecules that are closely associated with a ryanodine receptor, including CAM kinase, which is calcium sensitive. Now, not all the ryanodine receptors are in diets. That is especially true for large mammals, including humans. These are data for big ventricular myocytes. You see in green the staining of the plasma lemma and the T-tubules. And you see in red the staining of the ryanodine receptors and in yellow the co-localization. And you can appreciate that there are many ryanodine receptors that are not near a T-tubule. Actually, there are many fewer T-tubules in these large mammalian ventricular myocytes. So you have the situation on the right here. You have T-tubules and you have diads with ryanodine receptors that have the microdomain calcium. And then you have ryanodine receptors that have no T-tubules and perhaps they have lower values of calcium there. So we asked the question, if we now look at the point of view of ryanodine receptors, is there a difference between these different clusters? And do we see, because of the high calcium in the dyadic left, do we see calcium-dependent activation of CAM kinase in those groups of ryanodine receptors only? We know CAM kinase is activated in microdomains of high calcium. We also know it becomes activated during high-frequency stimulation. There's an integration of the calcium signal. So we had a rationale and a experimental testing model, and we set out to look at the response of ryanodine receptors in the diet, which we call coupled ryanodine receptors, and non-coupled ones that are sitting in between the T-tubules. And do they have a different response when we give high-frequency stimulation? To do so, we had to develop a much better way of identifying in situ during a functional experiment, coupled and uncoupled receptors without having to go to staining. This was much the work of Niall McQuaid when he was a postdoc in Leuven. And so here you see a staining of the myocyte with germ agglutinin to show the T-tubules. This is done in a 3D Z-stack and leading to a 3D Euclidean distance map. that then can be correlated with the actual events of calcium release on a single line during a single line scan through the middle of the cell. So what you're looking at here is the length of the cell, this is time, and the change in color is the rise in calcium. So this is the profile of the rise time, the 50% maximum value along the length of the line. So some are early, some are late. And this is correlated with the distance to the nearest membrane. And you can see that where you have late release, you have a long distance to the membrane, and where you have an early release, you are short to the membrane. And this translates in a T50 and distance relation, where you have the distance to the membrane and the T50. And so we said the sites that have this very early release are close to the membrane. This must be coupled clusters, and these are most likely non-coupled clusters. From that, we then define two groups to analyze. So we're not looking at the full line, we're just defined two groups. The coupled ones make up about 55% of the line scan, the non-coupled about 25%. So you see there's a group in the middle that we're not considering. We now can, along the line scan, based on the T50, we can say this is a coupled or a non-coupled site. We then, after conditioning the cell, we look at the spontaneous spark events that tell us about the properties of the random receptor clusters being coupled or non-coupled. To activate CAM kinase, we stimulated the cells at two frequencies. At 0.5, there should be no activation at CAM kinase. At 2 Hz, there should be strong activation. And we looked at the behavior of the RYR clusters. We see with an increase in frequency, we see an increase in spark frequency. This is the values in the coupled and the non-coupled. With the low frequency stimulation, this is with 2 Hz, both increase their frequency, but the coupled substantially more than the non-coupled ones. This is reversible, indicating it's presumably a reversible process, such as phosphorylation. It is CAM kinase dependent. When we did CAM kinase inhibition, we saw a decrease in the response in the coupled. The non-coupled were not affected at all, and now they ended up at the same value. Has it anything to do with CAM kinase being present in the dyadic cleft only? That's not the case. This is co-localization studies, random receptors, sodium calcium exchange for the membrane, CAM kinase. This is a triple staining, and we can then distinguish the association of the co-localization for coupled and uncoupled, and it was identical. Nevertheless, it is dependent on the structural organization, because if you disrupt the cytoskeleton, the modulation disappears. With other experiments, we have strong support that it is local calcium that activates CAM kinase. But it's not only CAM kinase, it's also local ROS production. Knox NADPH oxidase is localized at the sarcolemma, and the work of Ben Prosser and John Leder indicated it was involved in stretch-dependent modulation of IYRs. So we also looked whether it was in any way involved here. And inhibition of NOX2 equally decreased the excess modulation of the coupled IYRs without having any effect on the non-coupled. So we know from these experiments that dyadic calcium activates presumably CAM kinase through phosphorylation, but now there's also the possibility that it's actually oxidation through ROS from NOX2, and presumably there is an interaction between both, because they are both almost equally effective in suppressing this local modulation. Unfortunately, at this moment, we lack the tools to further investigate this relation. So lastly, a few words about the remodeling of microdomains with disease, which is the ongoing work in the lab. This is some work from Frank Heinzel a few years ago And we showed here that in a pig model of post-myocardial infarction remodeling, we have a reduction of T-tubule density. And when we look at the calcium transient, and this is again the confocal image length of the line, this is time, this is calcium release, you see it's quite irregular and lacks synchrony. And the areas of delayed release are the areas with low density of T-tubules, indicating we have a lot of non-coupled RYRs there. And indeed, the number of non-coupled is increased. This is also the case. This image is the same as what we find in human ventricular myocytes from heart failure patients. That was published a few years before by Bill Lauch. More recent data that did a more elegant study of the changes in T-tubules is from the group of Mark Hennell, who looked at the more detail of the organization of the T-tubules in human ventricular myocytes studied in short axis, and could show after heart failure disorganization and loss of T-tubules. So we have less coupled, more non-coupled, but do they still behave the same, or are there also now different microdomain modulations around these RYRs? So this is the ongoing work of Eve Dries. This is unpublished data. This is the pig model we are using. It's a non-surgical myocardial infarction induced by a stent that induces intima proliferation and high-grade stenosis. We have a lot of in vivo characterization, and that's a collaboration with the cardiac imaging group, where we can determine the in vivo function. So we have a moderate LV dysfunction. It's not a frank heart failure. We have a modest infarct, and we have areas of chronic ischemia around the infarct. So here is the scar. This is the area near to the infarct that is ischemic. And so we isolate myocytes from this area and compare to sham-operated animals. The myocytes are hypertrophied. They have less T-tubules, that is shown here on the right, and this is the increase in cell size. So I'd like to summarize here and give you back the overview of the work we have done and done by others. Calcium microdomains were a theoretical concept. I think the support for it from experimental data is huge, overwhelming, but we still do not have the direct calcium measurements. We know that the interaction in the diet is not limited to the L-type calcium channel and the ryanodine receptors, but it's also sodium-calcium exchange and the sodium homeostasis. There again, we need quantification. We also have now discovered and are really working on that to define better the functional signaling microdomains around RYR, and we're especially interested in what happens with disease, because clearly this affects these signaling microdomains. Where should we go? I think we need new tools. We need new probes targeted to the aortic cleft. And I'm very optimistic when I say they will measure calcium. We have first data. They are still not sufficient. There are still limitations in the current reporters, but I'm confident that this will follow. Also ongoing work is to determine the structure around these microdomains. What are the anchoring cues? What are the development and the signaling that determines whether or not we have T-tubules and we have more and less or less coupled and uncoupled RYRs. Importantly, this concept of microdomains is being used in other areas. It's being used to explain the influence of PMCA, so the plasma membrane calcium ATPases, strip channels, and others. And I think it's very important to take lessons from the work that has been done for the diadic cleft. It's important to get the functional evidence, to understand what calcium signals we are talking about, and to have some structural correlate to these microdomains. So to end, I think research, the work we do, is a combination of work and play. I think it's a privilege to do so. And it's been a privilege to work with many people who helped both on the work side and on the more pleasurable side of these activities. I first would like to thank the people in the lab, and in particular the current group and team, Eve Dries, who is still continuing the work in the myocardial infarction, she has gathered an enormous amount of data that has led to further insights and further questions. None of the work could have been done without the support of the cardiac imaging group, who provide the in vivo perspective to the things we do, and also in electrophysiology, but I didn't cover those data with Rick Willems. People who passed through the lab, very important. Virginie Bitour, she moved on to University of Hasselt. Frank Heinzel is now in Berlin. Carol Jaksait in Szeged. Gudrun Antons in Maastricht, and Niall went back to Glasgow University, whereas Lisbeth Biesmans chose for a job in the funding organization in Flanders. I would also like to express special thanks to a few people who have been long-time collaborators, supporters, and friends. David Eisner, who is never more than a phone call away when I have to discuss calcium and science. Mark Voss, who told me to keep an eye on translation and look towards the implementation of the work we did for treatment of arrhythmias. Barbara Casadei, who told me that there is more than calcium. There is reactive oxygen species, and there is NO, and there is NOx, and so on. And all three of them, I want to thank them for sharing with me there is more to life than work. Thank you very much for your attention.
Well, I'm still not convinced there's more than calcium, but still. Karin, I've read your papers over many years and particularly admired the originality of your work and the clarity of your thought, and I think everybody who's here today will have had a taster of that. So I won't go on at great length, but on behalf of the Physiological Society, I'd like to thank you very much for giving us such a beautiful Hodgkin Huxley Katz lecture this year. Thank you, and present to you with this.
Thank you.